Good afternoon. Welcome to today's Medical Center Hour. I'm Marcia Day Childress. I'm from the Center for Biomedical Ethics and Humanities, which is happy to bring you these weekly Medical Center Hour programs, often in partnership with other programs here at the Medical Center or Medical School and Health System. Today's program on the centenary of the 1918 flu, Remembering the Past and Planning for the Future, is a combined Medical Center Hour and Medical Grand Rounds for the express and excellent purpose of the Department of Medicine's annual Hayden-Farr Lecture in Epidemiology and Virology. This endowed lectureship highlights achievements in epidemiology and virology by esteemed physicians and scientists. The lectureship honors UVA Professor Emeritus Fred Hayden and the late professor and hospital epidemiologist Barry Farr. What a pleasure it is to welcome Dr. Hayden here today and to remember fondly our longtime colleague, Dr. Farr. This year's Hayden-Farr Lecturer is the distinguished pathologist Jeffery Taubenberger. Dr. Taubenberger is chief of the Viral Pathogenesis and Evolution section in the Laboratory of Infectious Diseases and deputy chief of said Laboratory of Infectious Diseases at the National Institute of Allergy and Infectious Diseases of the National Institutes of Health. He has done landmark work with the 1918 influenza virus and so is quite uniquely qualified to discuss with us past, present, and future concerns related to pandemic influenza. As you know, 2018 marks the centennial of the Spanish flu pandemic, the world's deadliest event, which killed at least 50 million persons worldwide. The pandemic's sudden emergence and high fatality are stark reminders of the threat that influenza has posed to human health and human society for more than a millennium. There's quite a lot we still don't understand about that pandemic, but the recent sequencing and reconstruction of the 1918 virus, work accomplished by Dr. Taubenberger and colleagues, represent a significant breakthrough. So how does this scientific advance help us better to know the past and prepare for the future? This fall, as you may know, Medical Center Hour has partnered with the School of Nursing's Bjoring Center for Nursing Historical Inquiry and Historical Collections in the Claude Moore Health Sciences Library, and now with the Department of Medicine, to observe the centennial of the 1918 flu. We're marking this somber anniversary with 3 programs, one examining the pandemic in the historical context of World War I, that happened in September, one focused on science, which is today's presentation, and one addressing the human toll of the epidemic. Program on the 14th of November, information about it is in your handout. So please see your handout not only for a bio sketch of Dr. Taubenberger, but also information about that final program 2 weeks from today. Also, please do visit the exhibit in the library lobby on the impact of the 1918 flu right here in Charlottesville, Albemarle, and at UVA. And if you've not already done so, get a flu shot. We'd like to thank the Department of Medicine and the Division of Infectious Disease for partnering with us today, and also all those involved in the Hayden-Farr Lecture. We also thank Historical Collections and the Library, sponsor of the History of the Health Sciences Lecture Series, of which this program is a part, and our Influenza 1918-2018 joint commemorative project with the university's historians of nursing and medicine. And now, Dr. Jeffery Taubenberger, and on the centenary of the 1918 flu. Welcome.
Thank you very much. Thank you, Marcia. It's an honor to be here and to give this talk on the 100th anniversary of the 1918 flu. So I will start with the important thing. I work for the US government and I can only dream of having something financially to disclose. So unfortunately I do not. It's an honor to give this combined talk in which I see that I'm supposed to talk about epidemiology, virology, medicine, and history, and I will do my best and show lots of slides and talk really fast and cover all those things. I didn't have the opportunity to meet Dr. Farr, unfortunately, but I have known and been friends and colleagues with Fred Hayden for over 20 years and can conclude that 20 years is a really long time, even for a good bottle of wine. So it's going to start with talking about the 1918 flu by thinking about last year's flu season, which was less than a year ago. I think you probably all recall what a really bad flu year we had. There were reports all over the nation about how bad the flu was, including here in Virginia. And the numbers of those impacted are just coming in and are looking to be very serious. It could be that as many as 1 million people were hospitalized in the United States for flu in the last flu season, and upwards of 70 or 80,000 people may have died. Of course, influenza comes in different forms, not only seasonal flu, but occasional pandemics in which a new strain gets into people. And it's here, especially on this day, that we think about the specter of pandemic flu and the worst one that we've ever seen, which one happened exactly 100 years ago in October 1918. So if we cast back 100 years and just pick one random example of what could be a description of what happened almost anywhere in the world in the fall of 1918. Here, Camp Devens, at the end of World War I, the United States was really gearing up to put men in the field on the Western Front. And Camp Devens was one of a couple dozen training camps in the United States for the Army. This was about 50 miles west of Boston. So here at the beginning of September, a single soldier presents with an influenza-like illness to the hospital. The next day, there were a dozen. A week later, there were 3 dozen, and a week after that, there were 12,000 U.S. soldiers in this one little training camp hospitalized for severe influenza. At the end of this outbreak, a third of the camp became ill, and nearly 800 men died. These were extremely healthy 18 to 25-year-old men who dropped dead of influenza. Here are some pictures of Camp Devens and their supportive care only for flu virus infections. They had no antivirals, no antibiotics, no vaccines, no intensive care, so all they could do was just generalized nursing care. A letter from one of the physicians treating patients that he sent to another one of his physician colleagues was not known at the time, but in the last couple decades was found. And this letter talks about how these patients develop the most viscous type of pneumonia that's ever been seen, and that you get these patients rapidly develop cyanosis and then struggle for air until they suffocate. It is horrible. We've been averaging about 100 deaths per day and keeping it up. William Henry Welch, who was probably the most famous physician in the United States at the time, was keenly involved in military medicine in World War I and came to tour this camp, and after visiting the camp was quoted to say that this must be some new kind of infection or plague, and yet it wasn't plague and it wasn't new, it was just influenza, a disease that had been recognized for at least 500 years before that. Here's some pictures of U.S. Troops being treated for influenza in a very primitive camp hospital on the Western Front in October 1918. So if we talk about some of the numbers, they're astounding, and I think that we will never know the full impact of the 1918 flu. The numbers keep rising as the developing world is looked at more closely. So currently, I think a reasonable number of deaths would be 50 million people in about a 9 or 10 month period in 1918, 1919, but 100 million is not unreasonable and is possible to be closer to the truth. In the US, there were a little under 700,000 people that died. So to try to put that in perspective, in a city like Philadelphia, 16,000 people died, 11,000 just in the month of October. So at its peak, 4,500 people died in Philadelphia in a 7-day period. The U.S. military entered World War I late compared to the European countries, but of 100,000 troops that died to all causes in World War I, 40% of them died of flu. There were about 17,000 deaths in Virginia, which was about 1% of the population at the time. Here's some work from a colleague at Michigan State, Dr. Chandra, who's been doing work as a demographer, trying to understand and model the influenza pandemic's mortality in countries where no mortality statistics were collected at the time by looking at the divot that occurred in the growth of the population. And from these work, he suggests that at least 14 million people died in India of the 1918 flu. Other estimates have gone as high as 17 million people. Here's a temporary camp hospital in Fort Riley, Kansas, again, where they recognized that it was a respiratory disease. They had patients wear masks and tried to separate them, but other than supportive care, there was nothing that could be done. The pandemic blew up and was recognizable everywhere in the world in September through November 1918 in a major wave. There seems to be some flu activity that occurred earlier in the late spring and early summer. Certainly in Northern Europe, there was clear evidence of the pandemic in June and July. But where the virus started and where it began to be circulated is not known and possibly will never be known because it's sort of under the radar of what happened. And then there were subsequent waves. Some places had outbreaks again in the early months of 1919. A very sort of sad and easy way to look at the impact of the 1918 virus is just to take a look at pictures of death records where each death record is a single page bound by month from the state of Oregon sitting on the shelf of the National Library of Medicine. And here you see the emergence of the flu in October, November, December, January, and then going back to a normal amount of monthly deaths for the state. You can find pictures not just in military camps, but in civilian life. Here's a picture of ill students in Dartmouth College, people in training camps, people at Walter Reed Army Hospital. You see Americans being Americans. They played baseball during the pandemic. They went to the theater. They had to have volunteers dig mass graves. This one gravestone, which is in central Pennsylvania, says, "In remembrance of the children buried in this area who died in the flu epidemic of 1918 and whose names are known only to God." This is not a site that you would expect to see in cities of the United States, but were relatively common sites in the fall of 1918. Its impact on World War I was tremendous. I think that the combatant countries could think of no good way to end the war, and I think the flu ended it for them. The leaders of the United States, Britain, and Germany all got the flu. Prime Minister Lloyd George almost died of flu. Woodrow Wilson became extremely ill with flu, and his slow recovery prevented him from participating fully in the peace conference, and that probably affected the way that the peace treaty was negotiated. General Pershing got the flu, Kaiser Wilhelm got the flu. Thinking about its impact on society, you can't emphasize, again, the enormous impact of the 1918 virus in terms of the personal tragedy of how many people died. Some of the most famous artists were affected. Edvard Munch in Norway got the flu but recovered, but almost died. Egon Schiele, the expressionist artist who was 28 at the time, died, and this is actually a picture of his deathbed. He died in the same bed with his wife, who was 6 months pregnant, they also all died within a couple days of each other in that same bed. Gustav Klimt also died of pneumonia in 1918. The impact of the virus was so great that it caused life expectancy in the United States to drop by about a dozen years. And this is due predominantly to one very remarkable feature which is still really unexplained, which is that usually influenza virus has its highest impact in the extremes of life, that is in neonates and in the elderly, two populations that are thought to be immunocompromised in some extent, with little mortality in between. So you get this roughly a U-shaped mortality curve for typical flu before and after. But in 1918, what you see was a new hump in the middle, peaking at about age 28. And this is the one cardinal feature epidemiologically of the 1918 virus that's seen everywhere in the world, that young, healthy adults had very high and unexpected mortality. And there are many hypotheses about them, but none of them adequately explain the data, and it still remains a big mystery. So let's talk very briefly about the biology of the virus itself. The 1918 virus was an influenza A virus. It's an orthomyxovirus. It's a single-stranded RNA virus with a segmented genome. Not to talk about the biology of the virus too much, but just to know that it's an envelope virus and expresses a couple surface proteins which play key roles in viral life cycle, one of them hemagglutinin, or HA, is involved in viral entry. It binds to the receptors on the cells, which are glycoprotein, the sugars on the tips of glycoproteins on epithelial cells. And it comes in a variety of subtypes or flavors. And the neuraminidase has the opposite effect. It helps newly formed viruses be released from the cell by cleaving off those same cleavages. The virus is an RNA, single-stranded RNA virus, and its polymerase lacks proofreading, so it has a lot of errors. So the virus lives in its kind of error-prone lifestyle where every single virion that's replicated has between 1 and 10 or more mutations. Many of these are deleterious, but the virus can rapidly select positively for mutations, for example, that help it escape from pre-existing immunity, from antiviral drug treatment, or if, for example, a virus from one animal finds itself in another non-native host, there might be some mutations that help select for adaptation to a new host. The kind of continual change in flu that we see, especially of the surface protein genes to escape pre-existing immunity, has been called antigenic drift. And because it's a segmented genome, if one host, and specifically one cell, is infected with 2 flu strains at the same time, you can mix and match the gene segments to create a completely novel virus that can have very different phenotypic properties, and that's been called antigenic shift. So you probably are all familiar with flu, but this is a helpful algorithm as we start in flu season to figure out if you have the flu. It's based on a lot of data. So it says, do you feel like you've been hit by a train, yes or no? No, you do not have the flu. Yes. Have you been hit by a train? Then yes, you've been hit by a train, and if no, then you have the flu. You can look at how rapidly influenza viruses change by doing sequence analysis in phylogenetic trees, and what you can see is that the influenza viruses that circulate in winter outbreaks every single year form distinct clades which can be dated, so that as if you look up a sequence in GenBank, you could say this human H3N2 virus is from 1998 or one in 1984, or so on. It's that rapid. You can actually look at molecular evolution in one host, and you can look at molecular evolution during one flu season. And most of that change is driven by changing the protein of the hemagglutinin on the top, where the antibodies recognize and where actually the hemagglutinin binds to the receptors that it's going to recognize. The result of this is that the flu vaccine has to keep up with this rapid evolution, and you can see the H3N2 viruses that have been the dominant flu strains in the last 50 years. The strain has to be changed almost every single year to keep up with this mutation rate, H1 slower. And so we clearly need to do a better job at coming up with ways to make vaccines that are not so sensitive to the kinds of mutations that flu viruses, to make more broadly protective vaccines, and I'll touch on that briefly at the end. So just to mention again, last year's flu season was particularly bad. It's estimated that it's possible, the numbers are not in yet, but it's possible that 80,000 people died of influenza pneumonia in the US last season, and almost 200 children were documented to have died. Influenza in our world is a human disease and is obviously a hugely important medical and public health problem, but influenza infections of humans is really kind of an accident. It's really a wild bird virus. It's a gastrointestinal virus in over 100 species of birds, but commonly in waterfowl like ducks and geese, shorebirds, terns, seagulls, etc., but many other species of birds have flu. So it's a GI virus. It's spread by oral fecal spread in the water. But influenza viruses have this ability to adapt to many other warm-blooded animal species, including other domestic poultry like chickens and turkeys, but also mammals, including wild mammals as divergent as whales and seals and bats, but important agricultural and economically important animals like horses, dogs, pigs, and of course to humans. And the complicated ecobiology of how influenza viruses move and adapt between these species is still only poorly understood, but means that eradication of the virus from humans is never going to be possible, like was possible for smallpox and could be possible for polio and measles. Flu is the poster child for Darwinian evolution and natural selection, with its rapid ability to respond to negative and positive selection. Here is a phylogenetic tree of the different subtypes of hemagglutinin, looking like a sketch that came out of Darwin's notebook. looking at the evolution of these different subtypes that occurred in birds. So if you were to read a textbook of influenza as to how pandemics form, you would see something like this paradigm, that birds like this duck are the natural host for flu, and that bird viruses adapt to pigs to become swine flu, and that swine viruses become human pandemics. So outside of Washington, D.C., there's very little evidence for this actually occurring, and it's just a much more complicated problem. It turns out that humans give viruses to pigs much more commonly than swine give viruses to us. In looking at this picture, I don't know which of the two is enjoying their kiss more, but they both seem to be enjoying it and sharing viruses probably in both directions. So if we go back and look at pandemics in history, we know of what's happened in the last 100 years. So we're talking about the 1918 flu, which was an H1N1 subtype, so that hemagglutinin subtype 1 and neuraminidase subtype 1. In 1957, there was a pandemic that was H2N2. In 1968, the Hong Kong flu, which is H3N2, which is still circulating. In 1977, the old lineage of H1 came back. And in 2009, there was a new H1N1 that emerged, and now this new H1N1 and this H3 virus continue to circulate as the influenza A viruses. We do not know what happened before 1918. If you look at mortality as just one measure of a pandemic, you find that pandemics are defined as having a novel virus, presumably an animal-derived influenza virus in completely or in parts, that has emerged in humans. But just because it's a novel virus does not mean that it's equally pathogenic or has equal public health impact. The 1918 flu had a huge impact, and the 2009 virus was a particularly mild pandemic, and yet it was a novel virus for humans. If you look at pandemics in history, certainly that they have gone back at least since about 1510. There have been 14 pandemics in the last 500 years, but there's some evidence of pandemics going back earlier than that, although it's very hard to look at the medical literature from the Middle Ages. So there were no virus isolates made in 1918. The concept of viruses was still rather new. While flu was recognized clinically, its causative agent was not. Spurred by the 1918 pandemic, researchers isolated the first influenza A viruses from pigs in 1930 and from humans in 1933. But by that point, there was no way to actually study the 1918 virus itself. So about 25 years ago at the Armed Forces Institute of Pathology, I had this crazy idea that we could perhaps use PCR-based approaches to find gene segments of the virus that caused the 1918 flu in autopsy tissues of people who died in the pandemic that were preserved in the National Tissue Repository. And this project worked, unfortunately or fortunately. Here are autopsy tissue sections cut and stained in 1918 that had been sitting on the shelf at that point for 80-something years and are still in good shape. And the tissues are a little bit of lung like this, fixed in formaldehyde and embedded in candle wax about the size of your fingernail. So using that, we had a small group of people that took 9 or 10 years to sequence the genome of the 1918 virus using what were, for us, pushing the technology as far as it could go back in the 90s. And then we expanded our collection of tissues going from other locations, including from a body that died of influenza, a person who died of influenza and whose body was buried in the permafrost in northern Alaska, exhumed by my colleague Johan Holten in Breivik Mission, Alaska. He did an exhumation in 1951 from the same site with the attempt to try to culture the 1918 flu virus, and that failed. There was no lab containment. They didn't even have biological safety cabinets in 1951, so I'm not sure what they would have done with the virus had they recovered it, but they did not. They looked at a place where in a local outbreak in a small Inuit village, 85% of the adult population died in 5 days and were buried in permafrost, leaving dozens and dozens of orphans with the same mortality pattern that young adults died and children did not. And it was extremely devastating to these communities. This is right on the tip of the Seward Peninsula of Alaska where Breivik Mission is. This is flying in about 10 years ago with the frozen Bering Sea here. And with apologies to Tina Fey, this is one of the very few places in Alaska you can see Russia from your house. This is where the bodies are buried on a bluff right on the beach in between these two wooden crosses. So if you look online, you see all sorts of weird things about how the 1918 virus was sequenced. And here, this thing says that we use these complex computer program and supercomputers to perfectly match the structure. So I just wanted to show you our supercomputer, which was that they were between femtogram and attogram amounts of viral RNA in the tissue. Most of it were single nucleotides. There was a tiny fraction of stuff that up to about 80 bases or so. You had to radioactively label the heck out of it to see anything, even with 40 cycles of PCR, and then do Sanger sequencing. So it took a couple of weeks to get 20 to 40 bases of sequence of the virus using this approach. And so a 10-year effort to put the virus together. So here's the supercomputer where we recorded the sequence of the 1918 flu. And then rebuilding the virus after the sequences were done was a multi-centered collaboration funded by the NIH that involved a bunch of different collaborators in a very exciting program project. And the 1918 virus could be resurrected to be studied in high containment labs to do model pathogenesis. Recently, we've been able to develop high throughput sequencing approaches that are much more efficient. And so now in about a week or two, I can sequence in my lab the complete genome of the 1918 virus instead of a 10-year effort, which is good because I'm old and impatient now. And we can do this more efficiently. So that means that we can look backward in time from 1918. Let me briefly show you some pathology from people who died in 1918. This is the histology of normal lung, which is mostly just blank airspace. And here what you see in 1918 deaths are features of the primary viral pneumonia with diffuse alveolar damage, with fulminant pulmonary edema and or hemorrhage, sometimes alveolitis with necrosis of alveolar cells, again, DAD, abundant thrombi. But the predominant pathology that you see was of secondary untreated bacterial pneumonias, massive bronchopneumonias with seas of neutrophils, destruction of the bronchial tree, but with evidence of repair going on, probably from the primary viral cause, and then death from the secondary bacterial pneumonia. You can see abundant bacteria by tissue gram stain, the usual kind of nasopharyngeal common causes of bacterial pneumonia, pneumococcus, group A strep, staph. But there were gram negatives and a variety of bacteria that caused secondary pneumonias in 1918. The influenza virus is present throughout the bronchial tree from nose to alveolus. Replicates in the tips of the cells lining the respiratory epithelium, not the full thickness epithelium, which is of interest. And we have cases going back to as early as May 11th, 1918, the earliest sequence of a soldier that died of the 1918 virus, and a partial sequence, and those sequences are identical with fall wave cases. So the most important lesson initially from sequencing the 1918 flu is that the 1918 virus is truly the mother of all pandemics, in the sense that every single human influenza infection, whether every year's seasonal flu for the last 100 years or the subsequent pandemics we've had in 1957, 68, and 2009, are all genetically descended from this one founder virus. So if 50 or 100 million people died of influenza in 1918, tens of millions of people have died of influenza in the last 100 years, all of them due by the successful introduction of a single virus into the human population. So we're 100 years into a pandemic era that has no signs of stopping. And so just to make this point, if you take the last 50 years of mortality, and including the pandemics that have occurred of the last 3 pandemics, 75% of the mortality has been to seasonal flu. So while there's a huge concern to prepare for and prevent pandemic flu, it's the annual flu that is causing most morbidity and mortality in the United States and the rest of the world, and something that we should think about very seriously. So now let me very briefly talk about what we've learned about why the 1918 virus had such an impact. And I'm going to talk about a variety of factors that come together to provide more serious illness in 1918 than in other influenza virus infections. So we'll start with the virus. You can work with the virus. It's a select agent once you have approval under very high containment conditions. And the 1918 virus is very pathogenic, unlike most human viruses, without adaptation in mice, in ferrets, in non-human primates. The virus basically causes severe disease and death very rapidly, and it's very different than what happens with normal seasonal viruses or other avian viruses. To make a long story short, all the genes encoded by the virus, the hemagglutinin surface protein gene is probably the major virulence factor of the virus, that just putting this gene on the backbone of a non-pathogenic modern human virus is enough to kill mice and ferrets. And this is not shared with the HA genes of other pandemic viruses. Again, to make a lot of data very compressed, we think that the 1918 virus is a very avian-like virus and that there was likely an avian ancestor to the pandemic shortly before the pandemic emerged, and that the 1918 hemagglutinin is a very avian-like hemagglutinin gene with just small adaptations to receptor binding. And we know that, interestingly now, and scarily, that the H1s of wild bird duck hemagglutinins that you can find in a pond just outside of Charlottesville, if put in the context of a mammalian replicating virus, share the same pathogenic features of the 1918 virus, so that the mutations that are not pandemic specific, but in a sense are just inherited from a bird H1. And since we know that H1 viruses share this pathogenicity, we wondered about the other subtypes that circulated in birds. So we made a series of viruses that were identical, but just differed in their hemagglutinin. All of these are hemagglutinin right out of wild birds with no adaptation. And all of them replicated in mouse lung, but most of them did not cause any appreciable disease, no weight loss. But some of them were very pathogenic, like 1918, with death in about a week. And it did not correlate with their ability to replicate in the lung. Some that were very pathogenic had lower titers, and some that were not pathogenic at all had higher titers. But the subtypes that were pathogenic were H1, 6, 7, 10, and 15. And they share a number of features in that they induce a very marked pro-inflammatory response. But they're very divergent on the family tree, and so it's not clear that they share structural features that account for this high pathogenicity, and that's something that we're still investigating. It's not just an animal artifact. If you take normal human bronchial epithelial airway cells, you can culture them in vitro, and you see that the viruses that are pathogenic in mice and ferrets are pathogenic, cytopathogenic in human cells, like H1 and H7. This is scary to me and others because some of these viruses have caused outbreaks in humans, and currently in the last 5 years in China there has been an H7 outbreak of an avian virus that has currently infected about 1,600 people and caused about 800 deaths. So let's talk briefly about host inflammatory factors. One thing that characterizes the 1918 and these other pathogenic bird viral infections is this very profound pro-inflammatory response that you see with an abundance of CD45 cells coming into the lung early on in infection, but especially crucial here is that 1918 viruses induce this huge neutrophilic infiltrate, which is very unusual for a viral infection, that they induce a lot of pro-inflammatory responses and cell death. So taking advantage of this, John Cash in the lab took a series of experiments in which he infected mice with a lethal dose of the 1918 virus, waited for them to be ill by day 3, and then treated them with a drug that is a superoxide dismutase catalase mimetic that reduces the production of free radical oxygens, has no antiviral properties, and yet mice that were injected with this drug for a week during the infection could actually clear virus and recover and have minimal damage in their lungs and little evidence of free radical oxygen damage, whereas untreated mice had this very profound death, suggesting that while the virus itself is a very virulent virus, it's not just the virus infection itself that's lethal, but the inflammatory response is contributing to an immunopathology. We'll talk briefly about bacterial factors. As I said, secondary bacterial pneumonias were a crucial feature in 1918. And as you recall, along the respiratory epithelium down through the bronchial tree into the lungs, the influenza virus replicates in the superficial cells, but not the basal cells, which also turn out to be the local respiratory epithelial stem cells, and rapidly reproliferate and recover and repair after a typical flu infection. But in the case of damage caused by a very pathogenic virus like 1918 and then secondary bacterial pneumonia, you get complete loss of these basal epithelial cells, and you actually lose the ability to reproliferate and repair. And I think this lack of repair is one of the reasons for severe disease and the progression of 1918 copathogenic pneumonias. Another interesting and still evolving story is that the inflammatory response generated by the 1918 virus with high levels of neutrophils actually ended up changing the behavior and gene expression patterns of secondary bacteria like pneumococcus strains here and actually make them more virulent. And in the case of the 1918 virus, what we think is that this coactivation of neutrophils by the 1918 virus and then subsequently by secondary bacterial infections induces a widespread vascular tree thrombotic picture. And if you go back to 1918 autopsies, you see very frequent small venule thrombi. And here you see massive factor III deposition in 1918 autopsies that you do not see in autopsies of people dying of the 2009 pandemic, even those with secondary strep infections. So this seems to be a 1918 unique thing that is inducing this thrombosis, and this may have certainly have contributed again to the extreme pathogenicity of the 1918 virus. Just as a quick aside, one of the cases showing a widespread thrombi had sickled red cells, and we ended up just doing PCR across the globin gene, and this was an African-American soldier who died in October 1918 who had the GLU6-Val sickle cell mutation. And so in a sense is the world's oldest diagnosed case of sickle cell anemia 4 years before the term was described in 1922. So studying influenza in animals is clearly very important, and it allows us to understand basic biology and pathogenesis of viruses like 1918, and certainly are crucial. But ultimately, as a physician, our goal is to understand influenza in humans and to control influenza in humans. And so we have been studying naturally infected patients with influenza in the last dozen years or so at the NIH hospital, concentrating on people who are at high risk groups, people who are immunocompromised, pregnant women, et cetera. But in the last 5 or so years, we have started doing studies that Dr. Hayden has done for many years in the past, and other groups had successfully done, but then people stopped doing them, which were volunteer challenge studies, in which healthy, very carefully screened volunteers are brought into the hospital and intentionally infected with circulating wild-type influenza viruses to study basic pathogenesis and immune correlates, but using this as a basis for phase 2 studies that are very efficient in terms of ability to look at efficacy of novel drugs or therapeutics and vaccines in small numbers of patients. So this has been something that's been done very successfully and safely with no adverse consequences for the last 5 or 6 years. Here's my colleague Matt Memoli, who runs all of our clinical studies. He's an infectious disease physician in my group, inoculating a patient with influenza using a little atomizer spray here. And this is done in a high containment suite at the NIH Clinical Center Hospital, the same suite where Ebola patients were treated. And we are actively recruiting for various studies all of the time and get a number of recruits from to help us with these studies, and that's extremely crucial. We've done over 400 patients so far with H1 and H3 viruses. We have additional H1 and 3 viruses and influenza B viruses in production. And we've done a number of phase 2 studies with novel vaccines and novel therapeutics that are helping us try to understand how to better prevent and treat influenza in humans. If one looks at antibodies in the blood, in the serum, as correlates of protection for flu, something that's been studied for decades and decades, most of the decision-making that occurs in terms of looking at people's protection against influenza as well as vaccine efficacy has been based on antibodies against the head of the hemagglutinin protein. So the antibodies that inhibit hemagglutinin from binding to its receptor, in this case in a surrogate assay, binding to red blood cells and agglutinating them in culture. And so this assay, hemagglutination inhibition test, HAI titers, are the marker that has been used for vaccine efficacy, and the idea that a dilution of this antibody to 1 to 40 or so is a protective titer. So what we've done in our challenge studies is to that it's difficult to do in a natural infected system is that we know when time 0 is for the infection, we know what their pre-existing titers are, we know the sequence of the virus, we can follow the development of their infection, their inflammatory response, and their subsequent immune response during the course of infection. And we also have been looking at other antibody titers as correlates. And interestingly, the hemagglutinin inhibition titer is clearly a correlate of protection, but it is not the best correlate of protection that we found. It turns out that the other surface protein, neuraminidase, is actually a better and an independent correlate of protection against prediction of shedding duration, symptom duration, the number of symptoms, and symptom severity. More recently, there's been a lot of emphasis in looking at antibodies against the stalk of the hemagglutinin that I'll talk about briefly. The part of the hemagglutinin that drifts less, that is under less mutation, and so the idea is that if you can make antibodies against this conserved part, you might be able to make a more universal vaccine. And stalk titers are also correlates of protection, but again, not as good an independent predictor of prevention of illness than neuraminidase antibodies. Here is some data from stalk antibodies. You find that rather than what some people have suggested, that stalk antibodies are actually rare, that we find that every patient that we've looked at has some detectable stalk antibodies in the serum to the 2009 virus, of course, over a wide range. But if you look, for example, at those who develop flu infection and develop illness after infection or inoculation in the challenge system versus those that do not, there is a difference in the mean titer in the stalk antibody. But importantly, that if you look at the highest quartile of stalk antibody, you find that about half the patients at this highest level of stalk antibody were in the group that was protected from developing infection and disease, and yet the people with stalk antibody titers at the same level still developed infection and disease, suggesting that it's not a perfect correlate of protection, and that while it certainly plays a role in protection, it might not be the magic bullet that some people would like it to be. If you look at influenza shedding and symptom development, you find that influenza viruses replicate often to a titer of as high as 10 to the 3 or 10 to the 4 in nasal wash fluid before the actual development of clinical symptoms, which is why influenza is so hard to control, because you can be shedding virus and transmitting virus to others before you really know that you're ill or very ill. So in this little window of the first couple of days, you have the possibility of trying to make some predictive biomarker assessments of who will develop more severe disease than others. And this is really important because at the moment, if someone presents with an influenza-like illness in the hospital, and even if a rapid diagnosis is made of influenza, it's not clear if this person's going to have a self-limited course, is this person going to develop a severe infection, is going to need to be hospitalized and given supportive care. And so if there were a way to assess this, this would be very useful. And so using our challenge system, since we know what time 0 is, and we have nasal wash and peripheral blood, serum, peripheral blood, white cell sampling every day during infection, we can begin to examine this. And by day 2, in studies of looking at over 100 patients, you can begin to look at gene expression in peripheral blood mononuclear cells on day 1 or day 2 that would predict duration of shedding. So you can make an annotated gene set that's very predictive of who will shed virus the longest. And you can do the same thing for symptom severity and how severe the illness is going to be. And so if these markers can be further validated in additional studies, that might be possible to provide some prognostic information to help guide whether patients might need more intensive therapy earlier on to try to prevent the development of severe illness, pneumonia, and death. So in the last couple of minutes, I will just go back to the problem that the current vaccine is based on an idea that we have to perfectly match the virus that is in the vaccine antigen to the virus that's circulating. Since flu viruses continually evolve and mutate in ways that are unpredictable, and that it takes at least 9 or 10 months from selection to making vaccine and filling vials and distributing it for vaccination, that we're always kind of behind the 8 ball, that we're always trying to catch up with flu evolution. And of course, as you know, sometimes the vaccine is a good match and sometimes it's a poor match. And last year was an example of a less than optimal match, and we had a very bad outbreak. Of course, we have no ability to predict when a pandemic will occur or what subtype it would be. And so at the moment, there's really no way to make a pre-pandemic vaccine. We can only make a vaccine against a virus after a pandemic starts, in which case it's really too late. In this day and age of interconnected travel, you know, it didn't take the SARS virus very long to be on 3 continents, and the same would happen with a transmissible novel flu virus. So there's been a huge push by my institute and others to try to make so-called universal vaccines, and the word universal vaccine could mean lots of different things to lots of different people. This could be perhaps a vaccine that would give you better breadth of protection from seasonal viruses so that maybe you don't need to be vaccinated every year. Maybe you only have to be vaccinated every 5 years or every 10 years. A broader one would be a vaccine that could actually be a pre-pandemic vaccine, that no matter what bird or horse or swine flu could get into people of any subtype, that you would have immunity. This is a pretty high bar given how diverse flu is and how much it mutates. I don't think it's possible to make a sterilizing vaccine that would prevent infection from any and all subtypes of flu, but I do think it might be possible to make vaccines that would at least help mitigate disease, reduce transmission, and reduce severe illness. And if you could actually prevent severe illness and death, that would be a huge public health improvement. So we have been, a number of groups have very interesting ideas out there, many of them based on stock antibody epitopes, but we've taken a more Jennerian kind of general approach, and we have been making a series of vaccines that are non-infectious that present a mixture of avian influenza virus hemagglutinins. These are the donor source for all human pandemic and seasonal viruses. And so as a proof of concept set of experiments that I'll show you, we make a non-infectious vaccine cocktail that expresses avian H1, 3, 5, and 7 in a variety of platforms. And we've been able to show in animals that it provides extremely broad protection against most or all influenza subtypes. So as an example, the avian virus could protect mice against lethal challenge against the 1918 virus. So this is within the same subtype that's in the vaccine, but not a matched vaccine to the 1918 virus, and yet you get 100% protection. But more importantly, and interestingly and intriguingly, we get protection against subtypes that are not in the vaccine. So the vaccine only contains H1, 3, 5, 7 hemagglutinins, and yet any other subtype that we can use to infect animals provides protection, for example, the 1957 H2 pandemic or an avian H10 virus, for example. So these are very encouraging results. And so in summary, in mice, what we have is that between 10 and 50 times lethal dose challenges with any subtype that causes disease in mice, we get 100% protection. You make antibodies against the vaccine antigens, but that does not explain the heterosubtypic protection because you do not make antibodies against the head of novel HAs you haven't seen. So there is a huge T-cell component to this as well, which we're investigating. And you can see that you get big rises in memory CD8 and effector CD4 cells. In the lungs, you get, here tetramer staining, you get a lot of flu-specific CD8 cells to various T-cell epitopes on the hemagglutinin in the head and the stalk. One of the features that induces severe pneumonias, like in 1918, is this influx of neutrophils in the part of the acute viral infection, and you completely abrogate and eliminate the influx of neutrophils in the lungs of mice and ferrets that receive vaccines, and you eliminate, here in ferret experiments, you eliminate the development of primary viral pneumonia and are just left with very mild sort of focal bronchiolitis. You reduce lung titers in ferrets by 4 to 5 logs. So these data are looking good, and we are in GMP manufacture of some candidates now, and we hope to be doing phase 1 studies in humans next year to be followed with small phase 2 studies in our challenge model in the future. So in summary, influenza is an incredibly complicated and protean problem. It has been with us for hundreds of years, and it likely will continue to be a huge problem. There is ultimately no way to eliminate influenza A viruses from human circulation unless we were to eliminate all warm-blooded animals from the planet, which I think would be bad. And so we have to cope with the fact that flu is here and continues to evolve and outwit us, how a virus with the 8 genes and 13,000 bases does a lot better than human beings. And we still actually don't understand very simple questions of how flu viruses move around between species, adapt between species, and vary in their pathogenicity. 1918 is an example of a virus that is helping us understand some of those questions, but is simply one virus out of thousands and tens of thousands and millions of flu viruses that have circulated and will circulate in the future. I think that it's crucially important to study influenza in humans. We need to do a better job understanding how humans are protected from influenza. And what we see is the incredible varied response I didn't have time to talk about, but that some people who are challenged develop big rises in their antibody titers after they clear their virus, and some people develop protection against their virus and their antibody titers never go up. So they're doing other things. Are they only doing a T-cell response? Are they developing stock antibodies but not head antibodies? And we're beginning to tease some of this out, and it's possible that there might not be a one-size-fits-all idea for new generations of vaccines. It might be that if you could work out the different kinds of responses that you have, there might be, in a sense, multiple vaccine choices available in kind of a personalized medicine approach in a very Star Trek-y kind of future look. So with that, I will stop and try to take some questions. But I want to acknowledge all the people in the group who've contributed to all the studies I've described, both in the group and our collaborators, and then particularly our funders. Of course, the NIH and the NIAID funding us, but we've also received extramural funding from DARPA, from BARDA, and from the Bill and Melinda Gates Foundation. So thank you for your attention, and I'd be happy to take any questions.
Wow, thank you very much. You're
welcome.
Taken us through much territory, and we have quite a nice amount of time in which to talk with members of the audience about what you think of this presentation and what questions you have. And Dr. Anthony Peters and I will have mics available to bring to you to ask your question or offer your comment. Please do identify yourself when you ask your question or offer your comment. So the floor is now yours.
Question.
All right.
Jack Gwaltney with a little bit of laryngitis, if you excuse me, but not a rhinovirus infection. You said what we think of the presentation. I thought it was wonderful, number one. When you challenge with your the volunteers with the spray, is that a small particle aerosol?
It is a large particle aerosol specifically designed so that we do not get lower respiratory tract involvement. So it's greater than 10 micron spray.
What about challenge with drops in the nose? Is the infection rate and the illness similar? Or?
You know, most of previous challenges have been used with drops, and we thought that perhaps a spray like this with a large particle aerosol would give better distribution, that there would be less chance of the inoculum sort of running out of the nostril. And, you know, we have not done a comparison of the technology. In our H1N1 series, if you have people who have low HAI titers before challenge, we see 75 or 80% of them shed virus and develop symptoms, so it's a very efficient system. In our recent H3 viruses, we're seeing a little less than that, about 60%, but still, this system seems to be a pretty good one. Of course, we want to prevent serious illness. Obviously, we do not want to induce a pneumonia in our volunteers.
There's some old rhinovirus studies that suggest drops give you a better infection rate than
That's
course
interesting.
aerosol. But did it appear that the deaths in the 1918 were mainly due to a secondary bacterial pneumonia? Is that what you said? Because you hear of these patients getting sick and dying so rapidly, it didn't seem like there was time for them to get a secondary bacterial pneumonia that would be severe enough to kill them. Mm-hmm. And I was an intern when they had the H2N2, and we did see patients that would come in and die very rapidly. And so I wondered what you thought about that.
No, that's great questions. I think that certainly that there were people who had very rapid courses, and there must have been some people who had, in a sense, kind of fatal primary viral infections. But if you look at the aggregate data, I think it's overwhelming that the vast majority of people died with secondary bacterial pneumonias. If you look at tens of thousands of published autopsy studies, including careful postmortem microbiology. 95% of them had culture-positive secondary bacterial pneumonias at death. The average course to death in 1918 from onset of illness to death was 11 days, exactly like the 2009 pandemic. So, certainly there were a tiny number of people who probably had very rapid deaths or cardiovascular deaths or other things. I think the clinical course of severe illness and death was one of viral infection, partial recovery, the secondary bacterial pneumonia, and then death 10 or 11 days later.
Your colleague, give it up after. Bob Chanak used to talk a lot about nasal antibody. Is that still part of the picture, or do you think there's anything to that?
That's a great, I could have paid you to ask that question. That's fantastic. As I said, I think that serum antibody correlates are clearly some marker of infection, but flu doesn't have a viremic phase, as of course you know. It's not a systemic infection, and you're looking at antibody at the wrong place, right? It's a localized mucosal infection. And I think the key to understanding why flu is such a problem maybe be why other mucosal-only infections are hard to develop vaccines against. Right? GI viruses or other respiratory viruses, be it RSV or paraflu or flu, is that there's something about the memory response in a mucosal system that's different from a virus like measles that has a systemic phase. So we think that in our group that looking at what's happening in the mucosal level is going to be really important. In all of our new challenge studies, we're going to be doing mucosal antibody, mucosal cytokine, mucosal brushings, looking at cellular responses as part of that. And we hope that this will help us guide vaccine development.
Jeff, thanks. That was tour de force.
Thanks.
Much appreciated. Oh, Fred Hayden. Two pathogenesis questions, if I may. In your autopsy work from 1918, did you look for extrapulmonary dissemination of virus? I know you've done some publication on whether there was virus in the central nervous system as an explanation for some of those syndromes, but I didn't know more broadly if you'd look. And then I have a second one after that.
Okay. We have not found any evidence of extrapulmonary replication of virus by PCR. And I think the pathology, the really good autopsies that were done at the time would really support that it was a respiratory-only thing. Of course, if you have a really bad fatal pneumonia and you're getting hypoxic and cyanotic, you have secondary hypoxic changes so that you would typically see, the kidneys and the brain and others to hypoxic damage, but not a primary viral thing. We do not think the virus was systemic.
Okay. No, that's helpful. There have been a number of reports, even with seasonal influenza B with myocarditis and things like that, leading to fairly rapid mortality in some unfortunate individuals. And then getting back to this issue of the importance of the host responses, these immunopathologic responses, and you showed data nicely on the reactive oxygen species. And have you examined any interventions that might influence the influx of polys or their activation? Have you had a chance to examine that in the animal models or in the challenge model? I mean, really, what should we be testing?
Mm-hmm. No, I think those are really excellent things. And that the neutrophilic influx is a really characteristic and kind of cardinal feature of the 1918 pathogenicity that seems to be shared with, say, H7 virus infections, not with H5 as an example. So it could be that for certain viruses with those subtypes that we've modeled, that pharmacotherapy that could in a sense influence neutrophil influx into the lung or activation, might be important. But there's a knife's edge, because if those people with severe flu infections develop secondary bacterial pneumonias, it's hard to know how you would do that in a clinical setting. So mostly we were trying to show that the inflammatory response itself was contributing to the pathology, but there is the possibility that you could intervene in a very careful way in a very severe viral infection in a way that could modulate immune responses, but we have not further investigated.
Kosti Sifri, infectious diseases. So I also want to thank you for providing just a fantastic lecture. I just was really curious specifically about your observation about neuraminidase and its marker as a correlate of immunity and in comparison to hemagglutinin, and sort of a two-part question that maybe settled science, but What do you think, is there any implication in terms of that finding with recombinant hemagglutinin vaccination, which is now on the market? And also, and this may be settled science, but is neuraminidase an alternative target for vaccine development?
I think that neuraminidase should be an important target for vaccine development, not necessarily by itself, although you could imagine, for example, that you could supplement neuraminidase in the vaccine. So a couple of points. The current vaccines, inactivated vaccines, are inactivated virus vaccines, but that then the hemagglutinin and neuraminidase are split off the virus and purified. The process is such that while neuraminidase is present in the vaccine, it's less likely to be immunogenic than the HA, probably because it's just less, the tetramer of neuraminidase is less stable. So often, even though neuraminidase is in the vaccine and it's not quantitated like HA, you variably get an immune response. Some vaccines do, some preps don't. And I think that making NA more immunogenic and quantitating NA would be a big improvement. So one possibility would be to have a supplement NA vaccine. We've done vaccine studies where we just make NA expressed on a viral-like particle, so no hemagglutinin, and we can vaccinate animals and provide really excellent protection against nasty viruses like H5. So clearly neuraminidase immunity is really important. These data are very old. I mean, their neuraminidase data go back to the 50s and 60s, that it's a really important correlate of protection. But for whatever reason, the sort of whole kind of government vaccine industrial complex for flu has just sort of focused on hemagglutinin titers in the absence of really good data. So I would strongly encourage that that new vaccine candidates, focus on adding at least immunogenic neuraminidase.
Eric Hewlett over here. Excuse me. You showed that virus shedding occurs before symptoms begin, but with the course of illness as a relatively brief period of time, how do you envision transmission occurring long distances to India and places like that in 1918 when they didn't have travel the same way that we do?
Yeah. That flu viruses are not nearly as transmissible as many other viruses like measles, for example, but they're clearly transmissible enough. So the R naught for flu is somewhere around 1.5, 1.7 or so. So one person can give it to 1.5 people or so. But that's clearly enough. And there's probably contact transmission. There's large particle and small particle aerosol transmission. I think one of the reasons that flu viruses transmit more in the winter months is that people are more likely to be closer together and indoors. Kids are back in school, these features. But, the spread of virus in 1918 or even before that was the same way. It's just person to person. And in 1918, the world was still relatively interconnected. That the exact same week that the flu peaked in Philadelphia, it was peaking in Auckland, New Zealand, and Cape Town, South Africa. I think the virus had been seeded everywhere in the world during the wrong time of the year. It was in the spring and the summer when it was the wrong time of the year for flu to circulate because temperature, humidity, and other things. And that when the appropriate time came up, it just exploded. So it really was everywhere. If you go back to the first pandemics of the 1500s that were seen and recorded in North America, they follow in less than a year from a pandemic in Europe and being brought by the tiny number of people coming in, the tiny number of ships coming to the New World in the 1500s. So clearly flu, spreads really well. There's a paper in the 1700s of a British physician who talked about how outbreaks can occur in towns in England faster than you can get there by coach. So, obviously that can't be true, but with this idea that they're seeded everywhere, and then when the right time comes, they kind of explode. That's, I think, the best explanation.
So we're out of time, but I'd like to thank Jeffery Taubenberger for an amazing hour. Thank you.
Thank you.